A multiple cloning site provides a number of sites for restriction enzymes to cut the plasmid. When the enzyme PVU2 digests the plasmid, for example, it produces blunt ends on the DNA. If the DNA with the protein coding region is also blunt-ended, it can be ligated into this plasmid. In this example, just upstream of the inserted DNA are 3 bases that remain from the multiple cloning site. CAG codes for the amino acid glutamine. Next, 6 histidine codons encode the peptide tag. Upstream are 2 more codons, one of which is a translation start site, ATG, which codes for methionine. Operator sites derived from the LAC operon control when the tagged protein is expressed. In the cell, repressor proteins bind to LAC-O, creating an obstruction for RNA polymerase. When an inducer is added to the culture medium, the inducer binds to the repressor proteins and allows RNA polymerase to pass through and transcribe the DNA into RNA. Another important region in the engineered plasmid is a ribosome binding site. The ribosome binds to the RNA at this site before locating the first AUG start codon and translating the RNA into protein. The ribosome continues translation until reaching a stop codon. When cells transformed with this plasmid are induced to express the tagged protein, the tagged protein becomes abundant in the cell, constituting up to 30% of the cellular protein. With many such cells growing in culture, a large amount of the tagged protein can be produced. The next step is to harvest the cells, break them open, and prepare a crude cell extract. A column, consisting of agarose beads attached to nickel atoms, can separate the tagged proteins from the rest of the cellular material. Within the column, the histidine tags on the proteins have affinity for and bind to the nickel-bearing agarose beads. The proteins that lack the tags do not stick to nickel and can be washed out of the column. Thus, the histidine-tagged proteins are separated from the others by a process generally called affinity purification. The tagged protein is eluted from the column. A low pH solution causes the histidine residues to become protonated and unable to bind to the nickel on the beads. Alternatively, a solution with a molecule called imidazole competes with histidine for nickel binding. In each case, the tagged proteins are released from the column.
This movie will guide you through the His-Mag-Sepharose nickel protocol. Note, this is only one of the many products available for mag-Sepharose. Before getting started, review the relevant instructions for use. For increased convenience, use the His Buffer Kit that includes stock solutions of all the buffers you will need. Start by mixing the medium slurry thoroughly by vortexing. Dispense 200 microliters of suspended magnetic beads into an Eppendorf tube and place it in the magnetic rack. Insert the magnet in the magnetic rack before removing the storage solution. Add 500 microliters of binding buffer with 5 to 60 millimolar imidazole. Immediately after equilibration, add 1,000 microliters of sample containing 5 to 60 millimolar imidazole. If the sample volume is less than 1,000 microliters, dilute the sample to 1,000 microliters with binding buffer. Resuspend the medium and incubate for 30 minutes with slow end-over-end mixing or by using a benchtop shaker. Add 500 microliters of binding buffer with 5 to 60 millimolar imidazole to remove unbound proteins. Repeat this step 3 times. Add 100 microliters of elution buffer. Resuspend the medium. Remove and collect the elution fraction. The collected elution fraction contains the purified protein. Mag-Sepharose magnetic beads have excellent properties for small-scale experiments. The Mag-Sepharose product range covers the following applications: enrichment of target proteins by immunoprecipitation or pull-down applications, capture and enrichment of phosphopeptides with titanium dioxide-based chromatography, high-capacity small-scale purification and screening of monoclonal and polyclonal antibodies from various species, and high-capacity purification and screening of histidine-tagged proteins from cell lysates. Mag-Sepharose, magnetic beads made easy.
After the tag protein elutes from the column, an enzyme is added to remove the tag from the amino terminal end of the protein. Another enzyme is added at the same time to modify the glutamine residue at the end of the tag. This modification prevents the digestion of the protein of interest. The enzymes used in this system have been engineered with histidine tags of their own. The tags allow the enzymes to be retained on a nickel-based column, while the protein of interest washes through. Finally, another enzyme removes the modified glutamine amino acid. This enzyme can be removed later from the mixture by using its histidine tag and the nickel-based column to separate the enzyme from the protein of interest. The steps involved in producing and purifying the protein of interest can be completed relatively quickly in the period of a week.